We're now going to consider the left wing of the catabolism diagram. I always like to start on the left rather than the right, for reasons I will not expound upon here. We're going to look at lipid catabolism starting with triacylglycerols. The triacylglycerols provide another source of energy for living cells. Quantitatively, triacylglycerols constitute the most important energy store in humans. Remember that carbohydrates stored as glycogen constitute less than 1 day's supply of energy. In contrast, the fats stored as triacylglycerols contain, on an average, enough energy to fuel basal metabolism in humans for 1 or 2 months. The general pattern of lipid metabolism is summarized on this slide. When energy is needed, lipid catabolism occurs. The triacylglycerols from the diet or from lipid stores can be broken down by lipases to fatty acids and glycerol. The fatty acid components of triacylglycerols are converted to acetyl-CoA by the beta oxidation pathway. The acetyl-CoA can enter the TCA cycle or can serve as a starting material for a number of biosynthetic pathways. The glycerol component is integrated into glycolysis or gluconeogenesis by a short series of reactions that feed into these pathways. On the anabolism side, lipid biosynthesis occurs when there's excess energy. The fatty acids can be synthesized from acetyl-CoA, and glycerol can be produced from glycolysis intermediates. The two components can be combined in ATP-dependent reactions to form triacylglycerols. Most dietary lipids are ingested as the triacylglycerol compounds. The dietary lipids are emulsified in the intestine by bile acids secreted from the gallbladder. Pancreatic lipases hydrolyze the triacylglycerols into free fatty acids and 2 monoglycerides in the lumen of the intestine. This diagram shows the pathway of dietary triacylglycerol digestion from the lumen of the intestine to the lymph system. The triacylglycerols are hydrolyzed into free fatty acids and 2 monoglycerides in the lumen of the intestine. The 2 monoglycerides and fatty acids are transported into the enterocytes, where they're reassembled into triacylglycerols and packaged into large chylomicron complexes. The chylomicrons are exported from the cell and transported in the lymph system to adipocyte cells. The adipocytes store the lipid as triacylglycerols until there's a need for energy. A photomicrograph of an adipocyte cell reveals that the lipids are stored in large vacuoles, which take up the majority of the cell volume. These vacuoles are so large that they sometimes displace the nucleus to the periphery of the cell. Another look at an adipocyte using a scanning electron microscope reveals the same spherical shape with a bulging nucleus on one side of the cell. In adipose tissue, the hydrolysis of stored triacylglycerol molecules is initiated by hormones such as adrenaline, glucagon, and ACTH. The triacylglycerol lipase is activated by an enzymatic cascade that's triggered by hormone binding to the appropriate receptor. After cleavage of the first fatty acid by the triacylglycerol lipase, the second and third fatty acids are also removed from the diacyl and monoacyl glycerol molecules. If this reaction takes place in adipose tissue, the fatty acids are released into the blood and are carried to other tissues in a complex that includes the blood protein albumin. Some of the glycerol is also released into the blood and can feed into glycolysis or gluconeogenesis in other tissues. The glycerol that's released from adipose tissue is taken up in other tissues, where it's integrated into glycolysis or gluconeogenesis by a two-step pathway. First, an ATP-dependent kinase reaction yields glycerol 3-phosphate. Then the glycerol 3-phosphate is oxidized to dihydroxyacetone phosphate by an NAD+ dependent dehydrogenase. This is a good time to distinguish between two terms that are used to designate functional groups that are derived from carboxylic acids. The term acetyl specifically refers to the 2-carbon group having a carbonyl and a methyl function. Acetyl groups can be viewed as derivatives of acetic acid. The term acyl is more broadly defined and encompasses a range of homologous carboxylic acid derivatives. This includes the acetyl group plus other carboxylic acid derivatives having one or many methylene groups. Fatty acids are carried in the blood from adipose tissue in association with serum albumin protein. The fatty acids are taken into the cytosol of target cells where they're activated by being converted to acyl-CoA derivatives. The conversion of a fatty acid into an acyl-CoA intermediate is a thermodynamically unfavorable process because the acyl-CoA is a thioester derivative. Thioesters have a very high energy of hydrolysis, which means it takes a lot of energy to make them, starting from a carboxylic acid. To make the acyl-CoA derivative, the fatty acid is first activated using ATP. The intermediates in the process are inorganic pyrophosphate and a fatty acyl adenylate. Pyrophosphate is cleaved to two inorganic phosphates with a release of energy, a pattern that we have previously noted, which contributes to the overall thermodynamic favorability of the reaction. The acyl adenylate derivative is a mixed acid anhydride with a high energy of hydrolysis. When it's swapped for CoA in the next step, the reaction is thermodynamically neutral. The formation of the acyl adenylate intermediate thus provides a way of using ATP to facilitate the synthesis of a high-energy CoA product from a free fatty acid and CoA. Fatty acyl-CoA derivatives are synthesized in the cytosol of the cell, but the oxidation of such derivatives takes place in the matrix of the mitochondria. An elaborate little dance must take place to get the fatty acyl-CoA into the mitochondrial matrix. In the cytosol, the activated CoA derivative is first transferred to carnitine, a polar carrier molecule. The fatty acyl carnitine derivative is then transported through the mitochondrial inner membrane into the matrix. In the matrix, the fatty acyl group is transferred from carnitine back to CoA. The transfer of a fatty acyl group from fatty acyl-CoA to carnitine occurs in the cytosol and is catalyzed by an acyl transferase enzyme. The reaction is energetically neutral and thus is reversible. The fatty acyl group is transferred from the thiol group of CoA to a hydroxyl group, noted in red, on carnitine. The overall carnitine cycle is outlined on this slide. The activated CoA derivative is first transferred to carnitine. The fatty acyl carnitine derivative is then transported through the mitochondrial membrane into the matrix. In the matrix, the fatty acyl group is transferred from carnitine back to CoA. Then the carnitine is transported back into the cytosol. The transporter that accomplishes this exchange process is an integral membrane protein. It's an antiporter that exchanges an acyl carnitine for a free carnitine. The acyl carnitine comes into the matrix, and the free carnitine goes back out into the cytosol on the same transport protein. One whole cycle of beta oxidation is illustrated on this slide. The process begins with a fatty acyl-CoA derivative with N carbon atoms, and it ends with a fatty acyl derivative with N-2 carbon atoms. The two missing carbon atoms are released as acetyl-CoA. These oxidation cycles are repeated, each cycle shortening the fatty acyl-CoA by 2 carbons and releasing an acetyl-CoA. In the final cycle, a 4-carbon acyl-CoA, butyryl-CoA, is converted to 2 molecules of acetyl-CoA. The beta oxidation process starts with an oxidation-reduction reaction. In the reaction catalyzed by acyl-CoA dehydrogenase, a saturated acyl-CoA is converted to an unsaturated product. The new double bond, which is in the trans configuration, is introduced between the alpha and beta carbons, coded in red. The cofactor in the reaction is FAD, which is reduced to FADH2. The second reaction involves the addition of water to the double bond, forming a hydroxyl group on the beta carbon. The reaction is catalyzed by enol-CoA hydratase. Note that the carbon carrying the hydroxyl group is now a new chiral center in the L form. In the third step, the hydroxyl group is oxidized to a ketone coupled to the reduction of NAD+ to NADH. The enzyme that catalyzes the reaction is L3-hydroxyacyl-CoA dehydrogenase. The final step in the cycle is catalyzed by beta-ketothiolase. In this reaction, a CoA molecule, coated in green, attacks the beta carbon of the fatty acyl-CoA. The fatty acyl-CoA loses 2 carbons, which are released as acetyl-CoA. The thiolytic cleavage is facilitated by the presence of the carbonyl group on the beta carbon of the acyl-CoA. That beta carbon becomes the site of attachment of the incoming CoA molecule to the shortened fatty acyl-CoA. The major fate of the acetyl-CoA from beta oxidation is entry into the TCA cycle. Because the acetyl-CoA is produced in the matrix of the mitochondria and is used by the TCA cycle in the same place, that process is facilitated. Here's another example of the potential of learning by analogy. The beta oxidation pathway and the TCA cycle both use common reactions that occur in the same sequence. That sequence is acyl-CoA dehydrogenase for beta oxidation versus succinate dehydrogenase for the TCA cycle. Both reactions use FAD as a cofactor and convert a single bond into a double bond. Enol-CoA hydratase versus fumarase. Both reactions add water to a double bond to produce a hydroxyl group. 3L-hydroxyacyl-CoA dehydrogenase versus L-malate dehydrogenase. Both reactions use NAD+ to convert a hydroxyl group to a ketone. The point is that nature has given students of biochemistry a gift here. You do not have to learn 2 completely different pathways. If you memorize the TCA cycle, then learning beta oxidation should be duck soup because it's essentially the same pathway with slightly different surrounding structures. The oxidation of fatty acids is a catabolic process which has the goal of producing energy. So the question arises, how much ATP can you get from the oxidation of a fatty acid? And as luck would have it, we're here to answer our own question. We can divide the answer into 2 parts. One cycle of beta oxidation yields 4 ATPs, 1.5 ATPs for the FADH2 and 2.5 ATPs for the NADH. In a round of beta oxidation, there's 1 acetyl-CoA produced. The acetyl-CoA enters the TCA cycle and yields 10 ATPs, 7.5 ATPs from the 3 NADHs, 1.5 ATPs from the FADH2, and 1 ATP equivalent from GTP. Let's look at the complete oxidation of a fatty acid to carbon dioxide. We arbitrarily pick stearic acid, which has 18 carbons, no double bonds, as the starting material. It costs 2 ATP equivalents to activate stearic acid to the acyl-CoA derivative. Remember that in the reaction, ATP is converted to AMP and inorganic pyrophosphate. That's the energy equivalent of 2 ATPs going to ADP and inorganic phosphate. Those 2 ATP equivalents constitute the only energy expended in the process. There are 8 rounds of beta oxidation, with each round producing 4 ATPs, so this phase yields a total of 32 ATPs. You might be tempted to think that there were 9 rounds of beta oxidation from a C18 fatty acid, but remember that the 8th round, starting with a 4-carbon acyl-CoA, produces 2 acetyl-CoAs. There are 9 acetyl-CoAs fed into the TCA cycle, and each one produces 10 ATPs for a total of 90 ATPs. Minus 2 plus 32 plus 90 gives you a net yield of 120 ATPs. Wow! Now you'll probably be asking another question, and if you don't ask, we'll ask it for you. That is, how do carbohydrates and fats compare as sources of energy? The answer is that fats are more energy-rich than carbohydrates. When we calculate the moles of ATP produced per gram, it turns out that glucose produces 0.17 mole per gram and stearate produces 0.42 mole per gram. That converts to 4 kilocalories per gram for glucose and 9 kilocalories per gram for stearate. In general, on a per gram basis, fats produce more than twice the energy as carbohydrates. This reflects the fact that fats are more highly reduced than carbohydrates, and a more reduced compound typically yields more energy in biological oxidation reactions. Up until now, we've only considered the beta oxidation of saturated fatty acids. There are some minor complications in the oxidation of unsaturated fatty acids. We'll use the oxidation of oleic acid, an 18-carbon fatty acid with one cis double bond, as an example. This fatty acid has its double bond between carbons 9 and 10. The first 3 rounds of beta oxidation proceed normally because all the bonds are saturated. However, on round 4, cis delta 3 enoyl-CoA is the substrate. The problem with cis delta 3 enoyl-CoA is that its cis delta 3 double bond should be a trans delta 2 in order for oxidation to continue. The enzyme enoyl-CoA isomerase makes that conversion, and from there on the beta oxidation can proceed normally. Because the oleate was already unsaturated, there'll be one less FADH2 produced than for the equivalent saturated fatty acid. The consequence is that the unsaturated compound will yield 1.5 less ATPs than its saturated analog. Another complication arises in the oxidation of linoleic acid, a C18 fatty acid which has 2 double bonds. The first double bond occurs between carbons 9 and 10 and is treated to the same isomerization sequence as shown on the previous slide. At the second double bond, a cis delta 4 enoyl-CoA is the substrate. The conversion of the cis delta 4 into the trans delta 2 bond takes 3 reactions. 2 reactions convert the cis delta 4 double bond to a trans delta 3. Then an enoyl-CoA isomerase converts the trans delta 3 double bond to a trans delta 2. And with that, we will leave fatty acid oxidation, and happy to do it. Most fatty acids contain an even number of carbon atoms and produce the 2-carbon compound acetyl-CoA as the only oxidation product. However, there are a significant number of fatty acids that contain an odd number of carbons. During the beta oxidation process, these odd-chain fatty acids produce acetyl-CoA until the last cycle. In the last cycle, the products are 1 molecule of acetyl-CoA and 1 molecule of the 3-carbon compound propionyl-CoA. There's a special metabolic pathway for the breakdown of propionyl-CoA. The propionyl-CoA is integrated into mainstream metabolism by conversion to an intermediate of the TCA cycle, succinyl-CoA. This process requires 3 metabolic steps. In the first step, propionyl-CoA is carboxylated by a biotin-dependent enzyme to yield D-methylmalonyl-CoA. The second reaction converts the carboxylated product from D to L-methylmalonyl-CoA. In the third reaction, a vitamin B12-dependent enzyme catalyzes the isomerization of L-methylmalonyl-CoA to succinyl-CoA. This figure shows the structure of coenzyme B12. The cofactor has a polyporphyrin ring system similar to that of the hemes, but the central metal ion in B12 is cobalt. Vitamin B12-dependent enzymes generally catalyze very exotic isomerization reactions in which carbon atoms are moved from one place to another. Ketone bodies are produced when there's a need for energy in various tissues, or when the rate of the TCA cycle is inadequate to utilize all of the acetyl-CoA produced by fatty acid oxidation. Under most conditions, acetyl-CoA is produced from glycolysis and fatty acid oxidation at a rate which can be accommodated by the TCA cycle. Under these circumstances, most of the acetyl-CoA is converted to carbon dioxide. The acetyl-CoA is diverted from the TCA cycle to the formation of ketone bodies, which are short-chain fatty acids. As a normal part of metabolism, when there's a specific need for extra energy in various tissues, such as heart muscle, the production of ketone bodies also occurs when the rate of the TCA cycle is inadequate to utilize all of the acetyl-CoA produced by fatty acid oxidation. Such conditions include starvation, uncontrolled diabetes, or as a result of some low-carbohydrate diets in which fatty acids are released very rapidly. There are 3 products of acetyl-CoA metabolism which are collectively referred to as ketone bodies. These metabolites are acetoacetate, beta-hydroxybutyrate, and acetone. In the first two steps of ketone body synthesis, 2 units of acetyl-CoA react to form acetoacetyl-CoA, and a third acetyl-CoA unit reacts to produce HMG-CoA. These are also the first 2 steps in cholesterol biosynthesis. In the third step of ketone body synthesis, an acetyl-CoA unit is then removed from HMG-CoA, releasing acetoacetate. The acetoacetate is converted enzymatically into beta-hydroxybutyrate and is decarboxylated non-enzymatically to give acetone. As we start our study of the synthesis of fatty acids, we take one last look at this chart of catabolism. We use this figure to remind you that every pathway has to release energy or it would not proceed in the forward direction. We've just looked at beta-oxidation of fatty acids, and we know that that catabolic pathway, which converts fatty acids to acetyl-CoA, releases energy. Now, some of the energy in fatty acids gets trapped as FADH2 and NADH, and those reduced nucleotides eventually yield ATP. However, in addition to forming reduced nucleotides, some energy is released as heat. That's the consequence of the pathway being thermodynamically favorable. That is, it has a negative delta G. Okay, so if the beta-oxidation pathway is thermodynamically favorable, as it has to be, and the pathway of fatty acid biosynthesis is also thermodynamically favorable, as it also has to be, and the 2 pathways connect the same 2 compounds, then logic suggests the 2 pathways have to have some differences. That is, there must be some way to introduce extra energy into the biosynthetic pathway so that it too is energy releasing, which it would not be if it were the exact reversal of the beta-oxidation pathway. Keep this in mind as we look at the biosynthesis of fatty acids. This table shows a number of differences between fatty acid biosynthesis and beta-oxidation. 3 of these differences contribute to the favorable thermodynamics of both pathways. The differences that change the energetics are: fatty acid synthesis involves a biotin-dependent carboxylation reaction, but oxidation does not. Malonic acid derivative is a participant in fatty acid biosynthesis, but not in beta-oxidation. All of the oxidation reduction reactions in fatty acid biosynthesis use NADPH, but one of the oxidation reduction steps in beta-oxidation uses FAD+. In addition to the energy-related differences, there are other contrasts between the 2 systems. As the name implies, beta-oxidation involves oxidation, while fatty acid synthesis involves reduction. The acyl carrier for beta-oxidation is coenzyme A, but for fatty acid synthesis, it's an acyl carrier protein. The hydroxyl intermediates in the 2 processes have a different chirality. Take another look at the beta-oxidation cycle. If we were to run the beta-oxidation cycle backwards, there would be a sequence in which acetyl-CoA condenses with an acyl-CoA to form a beta-ketone. The beta-ketone is reduced to a hydroxyl group by a dehydrogenase. The hydroxyl group is dehydrated to form a double bond by a dehydratase. The double bond is reduced to a single bond by a dehydrogenase. That's essentially the sequence which we will now see in fatty acid biosynthesis. The sequence of reactions for the beta-oxidation cycle running backwards is the same as the sequence for fatty acid biosynthesis running in the forward direction. In the biosynthesis process, malonyl acyl carrier protein, the replacement for acetyl-CoA, condenses with an acyl carrier protein derivative to form a beta-ketone. The beta-ketone is reduced to a hydroxyl group by a reductase. The hydroxyl group is dehydrated to form a double bond by a dehydratase. The double bond is reduced to a single bond by a reductase. On this slide, we compare the structure of CoA with the acyl carrier protein. The interesting thing is that the working ends of both molecules near the thiol groups are identical. At the opposite end, away from the thiol group, CoA has an ADP residue, whereas the acyl carrier protein has a small protein, molecular weight about 10,000 Daltons. The attachment of the protein portion is through a serine hydroxyl group. In the first step in fatty acid biosynthesis, acetyl-CoA is carboxylated to form malonyl-CoA. The reaction is catalyzed by acetyl-CoA carboxylase. The acetyl-CoA carboxylase enzyme contains a covalently bound coenzyme, biotin, which serves as a carrier of an activated bicarbonate molecule. The reaction takes place in two stages. In the first stage, bicarbonate becomes covalently attached to the coenzyme to form a carboxybiotin intermediate. The attachment of bicarbonate to the biotin coenzyme is coupled to the cleavage of ATP to ADP and inorganic phosphate. In the second stage of the reaction, the activated carboxyl group is transferred from biotin to acetyl-CoA to form malonyl-CoA. The formation of malonyl-CoA contributes toward making fatty acid biosynthesis an energy-releasing process. The carboxyl group that's added to acetyl-CoA to form malonyl-CoA will be released during a subsequent step in the biosynthetic pathway. That decarboxylation is an energy-releasing reaction, and it will help make the overall pathway thermodynamically favorable. The second step of fatty acid biosynthesis actually consists of two parallel reactions. In one of these reactions, an acetyl group is transferred from acetyl-CoA to an acyl carrier protein by an acetyl transacylase enzyme. In the other reaction, a malonyl group is transferred from malonyl-CoA to an acyl carrier protein by a malonyl transacylase enzyme. The products of these reactions, acetyl acyl carrier protein and malonyl acyl carrier protein, will react with each other in the next step. The complete cycle of fatty acid biosynthesis is outlined on this slide. There are 4 steps in this biosynthetic cycle, and as we saw before, these steps mimic the reversal of the beta oxidation cycle. In the first reaction, catalyzed by a condensing enzyme, malonyl acyl carrier protein reacts with acetyl acyl carrier protein to give a 4-carbon beta-keto acyl carrier protein product. There are a lot of things happening in this seemingly simple reaction. First, a 2-carbon compound and a 3-carbon compound are condensing with each other to form a 4-carbon compound. Wait, 2 and 3 equals 5, not 4. What happened to the 5th carbon atom? It turns out that in the process of forming the condensation product, a carbon dioxide was released from the malonyl acyl carrier protein. That not only accounts for the 5th carbon, but also explains where some of the energy comes from to make this reaction proceed toward product. The release of carbon dioxide is usually thermodynamically favorable, and this reaction is no exception to that rule. So carbon dioxide release helps to drive this reaction towards completion. That explains why the acetyl-CoA was carboxylated to malonyl-CoA in the first place, not to add an extra carbon to the pathway, but to provide some additional energy, giving the overall pathway a negative delta G. There's one additional feature in the first reaction that contributes to its favorable thermodynamics. In the process of forming the 4-carbon product, an acyl carrier protein molecule is released. That acyl carrier protein was attached to the acetyl group by a thioester bond, which has a high energy of hydrolysis. When the thioester bond is broken, energy is released as heat. Thus that bond cleavage contributes towards the negative delta G for the overall reaction. In the second reaction, the beta-ketone group is reduced to an alcohol with NADPH serving as the reductant. That reaction is catalyzed by beta-ketoacyl carrier protein reductase. The use of NADPH in this reaction is typical of what occurs in anabolic oxidation-reduction reactions. Remember that NADH is generally used to generate ATP, and NADPH is used as a reductant in biosynthetic reactions. The hydroxyl group that's produced in this reaction is in the D configuration. In beta oxidation cycle, the equivalent hydroxyl intermediate was of the L configuration. The third step involves a dehydration reaction. The elements of water are removed, creating a trans-carbon-carbon double bond. The enzyme catalyzing this reaction is 3-hydroxyl-acyl carrier protein dehydratase. In the final step of the cycle, enol acyl carrier protein reductase uses NADPH to reduce the double bond to a single bond. The product is a 4-carbon butyl acyl carrier protein. That 4-carbon acyl carrier protein can go on to condense with a malonyl acyl carrier protein to initiate a second round of biosynthesis, the product of which would be a 6-carbon acyl carrier protein. Each succeeding round of biosynthesis elongates the acyl carrier protein by 2 carbon atoms. Because fatty acid biosynthesis occurs 2 carbons at a time, most naturally occurring fatty acids have an even number of carbon atoms. For the most part, we're focusing on mammalian biochemistry in this course, but we'll now consider a process that does not take place in mammals. It's called the glyoxylate cycle. The glyoxylate cycle is an anabolic metabolic pathway occurring in plants and several microorganisms such as E. coli and yeast. This cycle enables organisms to use acetyl-CoA for the net synthesis of carbohydrates. It is absent in animals, including humans. Its absence explains why humans can synthesize glucose from pyruvate and TCA cycle intermediates, but not from acetyl-CoA. The glyoxylate cycle makes use of many of the reactions of the TCA cycle, but it adds 2 new reactions. Those 2 additional reactions are shown here in the middle of the complete TCA cycle. On this slide, we've removed the TCA cycle reactions, which are not involved in the glyoxylate cycle. The first reaction that's unique to the glyoxylate cycle is catalyzed by isocitrate lyase. That reaction cleaves isocitrate into glyoxylate and succinate. The second unique reaction is catalyzed by malate synthase. In that reaction, glyoxylate condenses with acetyl-CoA to produce malate. When we combine these 2 reactions with the other reactions of the TCA cycle, we get 2 4-carbon products, succinate and malate. And both of these products can go into the TCA cycle to produce any of the other 4 or 5 or 6 carbon intermediates. The genius of the glyoxylate pathway is that it provides a mechanism by which 2-carbon acetyl-CoA units can produce 4-carbon products. The glyoxylate cycle facilitates the net fixation of 2-carbon reactants into 4-carbon products, something that cannot be accomplished by the TCA cycle acting alone. Furthermore, one of the TCA cycle intermediates, oxaloacetate, can be used to produce phosphoenolpyruvate, and phosphoenolpyruvate can yield glucose via gluconeogenesis. The bottom line is that bacteria can make glucose from acetyl-CoA, and alas, humans can't. Mammals do not have the enzymes of the glyoxylate cycle. Without the 2 glyoxylate cycle enzymes, the TCA cycle absorbs the 2 carbons of acetyl-CoA and releases them both as carbon dioxide. There's no net fixation of the 2 carbons of acetate into 4-carbon products. That means there's something that can be achieved by lowly bacterium, but not by a mighty mammal. We and all other mammals are unable to carry out the net synthesis of TCA cycle intermediates from acetyl-CoA. Furthermore, and of extreme importance, is the fact that mammals cannot accomplish the net synthesis of glucose from acetyl-CoA. In fact, any compound that yields acetate or acetyl-CoA as its only product cannot be used as a net source of glucose. That can cause problems during fasting or starvation because the major energy storage compounds in mammals are triacylglycerols. Crucial levels of glucose must be maintained during starvation, but the fatty acids released from triacylglycerols cannot contribute to glucose synthesis. They can only produce acetyl-CoA. We now turn to a consideration of cholesterol, which is the good guy, bad guy of lipids. Cholesterol has many necessary functions, including serving as an essential membrane component and as a precursor of steroid hormones and bile acids. On the other hand, cholesterol esters that accumulate in the arteries of old guys like me are the villains which cause atherosclerosis, leading to heart attacks and strokes. The diagram shows that all of the carbons of cholesterol are derived from acetyl-CoA, the red carbons from the carboxyl group and the black carbons from the methyl group. Cholesterol has 3 6-membered rings and 1 5-membered ring, none of which are aromatic. This diagram breaks the pathway of cholesterol biosynthesis into 4 arbitrary stages. Although it's not shown here, each of those stages involves multiple steps catalyzed by many different enzymes. The first stage begins with the 2-carbon compound acetyl-CoA and ends with a 6-carbon intermediate, mevalonate. The second stage begins with mevalonate and ends with a 5-carbon activated isoprene analog, isopentenyl pyrophosphate. Next, in stage 3, 6 isopentenyl pyrophosphate units are polymerized to give a 30-carbon intermediate, squalene. And finally, in stage 4, squalene is converted to cholesterol by a series of rearrangements and ring closure reactions. We mentioned before that acetyl-CoA occurs at a branch point in metabolism. In this case, it serves as the precursor of cholesterol. In the first reaction, 2 molecules of acetyl-CoA condense to form acetoacetyl-CoA. The acetoacetyl-CoA can then react with a third molecule of acetyl-CoA to give 3-hydroxy-3-methylglutaryl-CoA. The thioester carbonyl group of 3-hydroxy-3-methylglutaryl-CoA is then reduced to an alcohol, yielding mevalonate. Notice that it takes 2 molecules of NADPH to convert the thioester, which is the equivalent of a carboxyl group, into an alcohol. That is a 4-carbon reduction, hence the need for 2 molecules of NADPH. In the process, a CoA molecule is released. The driving force for this series of reactions is the release of 2 CoA units and the conversion of 2 NADPH molecules to NADP+. In the next series of reactions, the mevalonate is first converted to a pyrophosphate derivative. That takes 2 ATPs going to ADP. The next reaction involves an ATP-dependent dehydration and decarboxylation. Wow, all that stuff happening in one reaction. I'm not going to burden you with the mechanism, primarily because I don't know how it works. Else I would happily burden you, and it would have very little effect on my conscience. The end result is a nice little 5-carbon compound, isopentenyl pyrophosphate, which has a carbon-carbon double bond at one end. Next, there's an isomerization of that new double bond to form dimethylallyl pyrophosphate. At that point, a 5-carbon dimethylallyl pyrophosphate molecule condenses with an isopentenyl pyrophosphate to form a 10-carbon product, geranyl pyrophosphate, with the release of an inorganic pyrophosphate. This series of reactions is energized by the cleavage of 3 molecules of ATP, a decarboxylation reaction, and the release of a pyrophosphate. I'm sure that the pyrophosphate is promptly cleaved into 2 inorganic phosphates to give the pathway a further boost. In the next reaction, a 10-carbon geranyl pyrophosphate condenses with a 5-carbon isopentenyl pyrophosphate to give a 15-carbon farnesyl pyrophosphate. An inorganic pyrophosphate molecule is released in the process, and its cleavage would again promote the forward reaction. This slide summarizes the rest of the pathway, and thank goodness most of the action takes place off the slide. I'm really sick of this pathway at this point. In essence, 2 15-carbon farnesyl pyrophosphate molecules condense to give a 30-carbon product, squalene. Then there's a series of reactions which bond various carbon atoms together to form 3 fused 6-membered rings and a fused 5-membered ring. Somewhere along the line, 3 carbons are lost as carbon dioxide. A double bond is introduced into one of the 6-membered rings, and one of the rings is hydroxylated. We're left with the final 27 carbons of cholesterol, 3 fused 6-membered rings, 1 fused 5-membered ring, and an 8-carbon hydrocarbon side chain. This final phase of the pathway is energized by NADPH oxidation, pyrophosphate release, and some other stuff which will remain anonymous. Here we see some of the good guy features of cholesterol. It's a necessary precursor of steroid hormones, including progesterone, testosterone, and estradiol. During my 8-month vacation each year, I teach Biochem 3021 over the internet from my home in Port Angeles, Washington. I love the town. It has a little bit of everything. Here's what Wikipedia has to say about Port Angeles. Port Angeles is a city in and the county seat of Clallam County, Washington, United States. The population was 19,038 in the 2010 census, making it the largest city on the Olympic Peninsula. The city is situated on the northern edge of the Olympic Peninsula, along the shore of the Strait of Juan de Fuca. It features a long, narrow sand spit named Edda's Hook that projects northeasterly nearly 3 miles into the strait, creating a large natural deep-water harbor shielded from the storms and swells that move predominantly eastward down the strait from the Pacific Ocean. The harbor is deep enough to provide anchorage for most kinds of ocean-going ships. The south shore of Vancouver Island and the city of Victoria, British Columbia, are visible across the strait to the north. Port Angeles is located in the rain shadow of the Olympic Mountains, which means the city gets significantly less rain than other areas of western Washington. The average annual precipitation total is approximately 25 inches compared to Seattle's 38 inches. Temperatures are heavily modified by the maritime location, with winter lows rarely below 25 degrees and summer highs rarely above 80 degrees. However, in winter the city can be vulnerable to windstorms and Arctic cold fronts that sweep across the Straits of Juan de Fuca. Port Angeles receives about 4 inches of snow each year, but it rarely stays on the ground for long. Port Angeles is also the location and headquarters of Olympic National Park, which encompasses most of the Olympic Peninsula. It was established by President Franklin D. Roosevelt in 1938, just 5 years before I was born. This is a view of Port Angeles from the harbor. The town is wedged between the ocean and the Olympic Mountains. There's a daily ferry service from the port to the town of Victoria on Vancouver Island. The Coho Ferry crosses the Strait of Juan de Fuca in about 90 minutes. One of the major industries of the town is fishing. The port is home to many commercial and recreational fishing boats. The Lillian S. is probably older than I am, at least I hope it is. Peabody Creek runs through a